y vamos a dar comienzo a la sexta sesión de conferencias. Vamos justitos de tiempo, entonces, de entrada, pues solicitar a los ponentes una brevedad y ceñirnos un poquito al tiempo correspondiente. Va a empezar la sesión con la intervención, que para mí es un honor presentar, la intervención del doctor Francesco Paolocci. Su situación, su trabajo actual está como investigador dentro del Consejo Nacional de la Recherche del Instituto de Genética de Plantas en Perugia. Hizo su grado en agricultura en el 89 en la Universidad de Perugia y empezó a trabajar en el CNR, también en Perugia, en el 1990. Un joven investigador, pero con mucha experiencia, que en el 92 marchó a Australia, en el Siro de Australia, en Canberra, durante 2 años estudiando metabolitos secundarios de plantas. Su posición permanente en el CNR es a partir de 2001 y desde 2006 a 2010 también imparte docencia en la Universidad de Urbino, en la Facultad de Ciencias y Tecnología. Su investigación actual se expande desde estudios de genética y control medioambiental y en biosíntesis de metabolitos secundarios en genética también, así como en el ciclo biológico, ciclo vital y ecología de distintas especies de trufas. Trabaja con trufas desde 1994 y pues lidera, es team leader de varios proyectos en el CNR de Perugia igualmente, y finalmente, bueno, por decir que tiene más de 50 papers indexados a nivel internacional en las mejores revistas mundiales. Colabora también con el grupo de Nancy, una colaboración intensa, y en definitiva se trata de uno de los grupos más interesantes y de mayor repercusión que tenemos ahora mismo en orden del ciclo biológico de las trufas. Vamos a aprender muchísimo, yo creo, que con la presentación de Francesco, al cual pues, le agradecemos ya la venida aquí. Muchas gracias.
So, buenos días. Muchas gracias for your nice presentation. And I would like, first of all, to thank the organizer for inviting me to give this talk, and so for giving me the opportunity to attend this fruitful conference, and also for the opportunity to see the Silicon Valley data trufa yesterday. You know, it was amazing. So, my talk is about the sexuality of truffles, so life cycle of truffles. And as you heard during this conference, you know, many speakers talk about distribution of mating types in the soil and so on, and I'm really impressed by the relevance of our work as having on truffle and truffle nursery and so on. I would say more outside Italy than in Italy, indeed. So today I will give you an overview on the step we followed, we performed to unearth the truffle life cycle and the sexual reproduction. And then I will give you some of our last results about truffle sexuality. So we start approaching the truffle life cycle more than 15 years ago. So at that time, the leading view was the truffles do not need a sexual partner to complete their life cycle and so to fruit. And the leading view was that truffle mycorrhiza were eukaryotic and free bodies developed from these dikaryotic active macrolides. Indeed, if you look at the pattern of the hyphae, you can see multiple nuclei in the same cells. Some could resemble dikaryotic structures. Now, in this cartoon here, we have like a very simple representation of what we expect in the case of selfing or outcrossing by using codominant molecular marker like simple sequence repeats, also known as macrosatellites. Briefly, if you are in outcrossing species, you will see heterozygous patterns. I mean, you will see 2 alleles per each locus, okay? If you are working with dikaryotic structure, and this dikaryotic structure, is formed by different strains, genetically different strains, you also will see the heterozygous patterns. However, Berthaud and colleagues in 1998 published a paper where they first to use microsatellite markers, called codominant markers, to type tuberanosporum ascocaps, and they didn't see any patterns of heterozygosity. So they conclude that tuberanosporum was homotypic or even exclusive species. And this was the leading view has been the leading view for many, many years. So we start reconsidering the truffle life cycles when we perform analysis on the genetic diversity of tubermagnatum. So now we shift from tuberanosporum to tubermagnatum. A few years ago, we ran analysis to study the genetic diversity of natural population of tubermagnatum. We genotyped more than 300 samples distributed into 26 populations in Italy and in Eastern Peninsula, using 8 microsatellite loci, polymorphic loci, published in this paper. Now, this slide shows that we can differentiate some population according to these 8 markers. But the most relevant point, at least for what concerns today's talk, is about the population genetic analysis we performed. Basically, we analyze for the presence or not of linkage disequilibrium. Briefly, linkage disequilibrium is index that allow us to make inference about the reproduction. We have a significant linkage disequilibrium if we don't have any genetic change between individuals. Now, if we consider all these truffles as belonging to a single population and you run linkage disequilibrium analysis, you will see you have a significant linkage disequilibrium. This means that genetic exchange does not occur between individuals. And this could be due to selfing and or geographic barriers. Then we considered all the individuals within each single population. And we ran, once again, linkage disequilibrium analysis. In this case, we didn't have significant linkage disequilibrium analysis. So that means we were in front of linkage equilibrium. So we are in front of gene flow. So this population genetic analysis allow us to conclude that individuals within the same population, also between geographically close population, can change their genetic material. So this is a preliminary hint in favor of the crossing. So at this stage, we were in front of 2 sets of results. One suggesting the absence, so the selfing, the absence of crossing. The other on tubermagnatum suggesting the occurrence of events of the crossing. So now, which one was the right one? Sorry. So we realized that the first set of results suffered indeed from a methodological bias. Now, we have a tubermagnatum ascocarp. That could be also tubermagnatum, it doesn't really matter. So if you look at this ascocarp at the microscope, you will see the gleba, the asci, and the spores. If you look after this structure after grinding the material for DNA isolation, you will see that the asci and the spore are undamaged. So the DNA contributed by this ascocarp is only contributed by the gleba. So we decided to check for the DNA inside the spores, okay? So we isolate the spores from the surrounding gleba. We set the protocol to disrupt the spores and to isolate DNA from the spores and from the asci. So by so doing, for each truffle, we have two pools of DNA, one contributed by the gleba, the other contributed by the Ashai, to perform codominant molecular analysis. Okay, this is a sample for 500, tuber magnatum. We amplify, using this locus, the gleba. As you can see, we have only single peak. And this is expected. This is consistent with results produced by Berthaud and colleagues in tuber spore. When we analyzed the DNA contributed by the Ashai, we saw two peaks. So this is consistent with heterozygosity, with outcrossing. So we interpret this result to mean that the gleba is of uniparental origin and is made up of homokaryotic hyphen. Whereas in the Ashai, the additional allele we see with respect to that present in the gleba has to be provided by a sexual partner. So this approach allows us to provide the first evidence of the crossing to the magnatum. We repeat this experiment with analysis using several loci on different samples. In many, but not in all samples, we were able to find additional leads in the Ashai with respect to the gleba. Then we isolate polymorphic microsatellite in tuberous sporum, and we use the same procedures for DNA isolation in tuberous sporum, and we had the same result. So this is the gleba, and this is the Ashai. So the Ashai, we have two peaks, the gleba, we have only one. So once again, we have provides, we think, compelling evidence support that this fluid body results from a crossing. However, as I mentioned you earlier, only the gleba and the Ashai from a few subsamples of tuber spore fluid body show different SS and patterns. So we were able to provide compelling evidence of the crossing only in a subsample for a subsample tuber sporum. What about the others? So the question was, now, are truffles obligatory or facultative outcrossing species? Okay, we know that fungi can be divided in two main categories, homothallic fungi and heterothallic fungi. Homothallic fungi can self-fertilize, but also can outcross. Heterothallic fungi cannot self-fertilize. They have to outcross. And the locus that governs this reproductive mode is the mating type locus. In Ascomacetus, within the mating type locus, there are two main genes. They mate 1-1 and they mate 1-2. For simplicity, mate 1 and mate 2. Both code for transcription factors. One for transcription factor containing an alpha box, and the other containing HMG box domain. Now, in homothallic fungi, both genes are harbored in the same strain. So this strain has all the genetic repertoire to fruit, okay? So this strain can opt to outcross or to self-fertilize. In heterothallic fungi, we have different situation. These genes are harbored by different strains. So we have two kinds of strains, let's say minus and plus strains. So these fungi cannot self-fertilize. So our results were consistent with both, because we show the presence of crossing in some samples, but we were not able to show outcrossing for all, okay? To answer to this question, we have to look at the configuration, structure, organization of mating type genes. Mating type genes are not so well conserved across fungi. And at this time, Francesco Martina invited us to join the consortium for the sequencing of tuberanosporum genome, and our task was to identify the mating type genes in the sequenced genome. Okay, so we scanned the genome and we found only a single mating type gene. So the mating type gene that code for HMG box containing transcription factor. What about the other one? To search for the other one, we designed primers on this gene, and we screened some fruit bodies, tuberanosporum fruit bodies. As you can see, only tuberanosporum fruit body show the aspect amplicum for this gene. So we use a stratagem to clone the second one. I'm not going now to give you the details, quite boring, but using like a PCR-based strategies, we have been able to clone the second mating type gene on those samples that do not harbor this mating type gene, and this is the result. So this mating type gene is present in those ascocarps that do not have the other mating type gene. This is alignment of the met locus in two strains, one containing the MET2, the other containing the MET1 gene. So as you can see, now we can say that tuberanosporum is heterothallic because we select many strains, we check many strains, and all strains either harbor this or this other mating type genes. Okay. As is, so there are many points that we could arise looking this. So we talk about a lot about organization structure and so of the mating type locus in the tuberanosporum, but I don't have time today. And you know, it has been published, so don't worry. I mean, you can check the papers. I just would like to stress the fact that the mating type genes are not so well conserved across fungi. So this alignment, aminoacidic alignment of the alpha box domain, so the most conserved domain, among tuberanosporum and other ascomycetes. As you can see, this domain is poorly conserved. Let's keep this one. So we provide evidence, clear evidence that tuberanosporum is heterothallic. Now, this findings begs for few questions. I outlined only 3 questions here. Are other tuber species heterothallic as well? We can say that the tuber species we have screened thus far are indeed heterothallic. We have collaboration with Claude, Murat, and Francis Martin for this analysis. And we are confident that also other truffle species are heterothallic as well. Then the next question is, so what are the genes related to mutual recognition? So we have strains plus, let's say, and strains minus, okay? So how can they recognize each other? We know from other ascomycetes that, for instance, from Saccharomyces cerevisiae, that strains of different mating types secrete different pheromones, and the pheromones mediate the mutual recognition between strains of different mating types. So we annotated some genes in the tuberanosporum genomes relative putative candidate genes to the production by synthesis of pheromones, of pheromone receptors, and also to the pheromone signaling transduction pathway. And if you like to have more information, I invite you to see our poster in the poster room by Beatrice Belfiore, our group. And then the third question is how strains of opposite mating type are distributed. So to address this point, we initially screen a natural truffle grounds located in central Italy. Okay, so this is a truffle owner. When we saw him, okay, we are going to sample, to make some sample in your truffle to search for the truffle sex, he thought we were crazy. However, in this truffle ground, we sampled soil, and we analyzed the soil for the presence of mating type genes, for the presence of strains of different mating type. We analyzed mycorrhiza and the fruit bodies, and we genotype these structures by macrocellular light and mating type genes. So, now, this table shows the results we obtained analyzing the ectomycorrhiza. We sample ectomycorrhiza below some plants here. Briefly, as you can see, we sample many ectomycorrhiza, but all the ectomycorrhiza below the same plant always showed the same mating type, and also the same multi-locus profile at 2, 4, 6, 8, 7 SSRL loci. So this results allow us to argue that single plant in this natural truffle grounds hosts a single strain. So, and this, of course, says that the distribution of the two mating types in a single plant is biased. So overall, we have been able to identify 7 genesis, 7, let's say, strains in these truffle grounds, okay? 2 with a metin type plus and the other with metin type minus. And as you can see in this part of the picture, there's also a skewed distribution of the 2 metin type in the field. We have some area of the truffle ground where we could find only metin type minus ectomycorrhiza, some others where we found only metin type plus mycorrhiza. For the soil, it was quite different because some soil samples showed both metin types. And then also we compare the genetic profiles of ascocarps with the ectomycorrhiza collected nearby. This is our 2 examples. So this is ascocarp collected site 243. The ascocarp present the gleba with a strain plus, and this G is the pattern of the gleba, SP is the pattern at the spores. As you can see, the ectomycorrhiza collect next to the fluid bodies shows the same metin type of the gleba and the same multilocus profiles. Here, for this, but also for this, where we have the gleba minus, the ectomycorrhiza formed by mycelia with metin type minus. Again, the genetic profile of the gleba was identical to the genetic profile of the of the gleba, of the fluid bodies. So this led us to argue that ectomycorrhiza are haploid, so are homokaryotic, because you can see only a single allele per locus, and serve as a maternal partner in the cross, because the gleba shows the same genotypes of the mycorrhiza collected nearby in both cases. And also, it seems like politically correct, both metin type plus and metin type minus serve as maternal partners. So they share their job. Then, we have, if you look at the spores, the alleles present in the spores, okay, you can assume the genetic profiles of the male partners. And so we're combining this information, you will see that we have additional genotypes with respect to the genotypes we detect at mycorrhizal level. Okay? So this could be interpreted in 2 different ways. One, we didn't sample all mycorrhiza, so all the biodiversity at the mycorrhizal level present in the field, or that the sexual partner, so the male partner, is erratic. So it's not present at root resin strain in the truffle ground. We went further and we studied the presence of the 2 metin types genes on artificially inoculated host plants. So we used 4 truffles, 2 with a gleba minus and 2 with a gleba plus, to inoculate under control condition, nursery condition, we say, plants, and check the mycorrhiza. We check the mycorrhiza after 6 months from the inoculation, and as you can see, all the plant shows basically the same unequal representation of both metin type, let's say. The situation changed a little bit, or let's say more, it changed a lot, 1.5 years from the inoculation. As you can see here, some of the plants show a skewed representation of 2 metin types on their roots, but most of the plants showed only a single metin type. So this tells us that it cannot be taken for granted that spore inoculation ensures you the presence of both metin type on the host plant. But also, we interpret this to mean that interspecific competition phenomena are occurring on host plants. And this interspecific competition phenomena could be mediated by, or could be related by, the configuration of the metin type of locus. So media like lots of these results, so they start talking about sex war of the truffle grounds. So is it really a sex war? We don't know. But to deep our insight on this, we decide to check what's going on also on artificially truffle grounds. So we ran a parallel analysis in Italy and in France. In France, thanks to the collaboration of Claude Murat and Francis Martin's group. So in Italy, we analyzed a truffle orchard in central Italy using 12 microsatellite markers and mating type genes. We screened the mycorrhiza and the soil sample. So blue X means ectomycorrhiza plus. Sorry, it's not clear here. It's green, mycorrhiza minus, and the dark is no mycorrhiza. And this for the soil, the mating type of the soil. Briefly, we sample mycorrhiza all around this plant. As you can see here, all around this plant, we have only a mating type and only a single genet. Here we have 2 genets, but only a single mating type. Here we have 3 genets, but all share the same mating type. In this case, it was a mating type minus. In the soil, you can find both. Here we have, again, 1 mating types, but 2 different genets. So overall, we found 8 genets at mycorrhizal level, and we also measured the size of the genet was 2.3 meters. Claude led the study in Rolandville in France, and I think he is going to show you this morning some of these results. So I don't have time to explain much, but I think just to show that also the results from Rolandville truffle orchard reinforce our contention that the distributional pattern of a mycorrhiza is related to allelic configuration of the mating type locus. This is the results we obtained running GeneLand, and let's say GeneLand subdivided all the genets in 2 main groups, and these 2 main groups are related to the configuration of mating type locus. So now we can talk a little bit about practical relevance of our findings. So we think the bias distribution of the 2 mating type might limit the truffle production. So the question is, how can Romeo reach its Juliet? So the strategy that we can perform to supply a sexual partner to root resident strain. One will be spore dissemination, like a farmer that sows seeds. And we know that truffle growers used to do that long before we discovered the truffle life cycles. One other will be soil management. Soil management could increase the fertility of the soil, but also could increase the chance of diffusing meiospore or mitospore. We know now that truffle also produces mitospore. No? I think we learned more this morning. Another opportunity is to outplant next to plants that show only a single mating type, seedlings harboring the second mating type to supply a sexual partner. So we think it is time now to reconsider also the approach to establish ex novo plantations. So why don't we, our goal, and we are going to do this, to study the condition that optimize the chance of mating between strains of different mating types. And they could be realized following a scheme like this one by outplanting close to other seedlings harboring different mating types on their roots. And this could be done if we could certify plants according to their mating type. So now I'm trying to be visionary or foolish, okay, but I think not crazy. And so we can use seedlings, we can inoculate seedlings by using in vitro grown truffle mycelia of known mating type to produce lots of plants of known mating type to outplant. And we are now testing this approach in experimental field in Italy. So, in summary, thanks to microsatellite and mating type-based marker, we have been able to revise the life cycle of tuber melanosporum. So these are spores. The spores are plurinucleated, but they are all genetically identical because we test this. And also we have segregation of mating type genes between the spores. The spores are germinated in soils that colonize the roots and they develop a mycorrhiza. Mycorrhiza serve as a maternal partner in the cross. Who is the paternal partner? Would be another mycorrhiza strain, but would be a meiospore or mitospore that fertilize. Fertilization is So it means the eukaryotic phase. Indeed, we are not able to find any eukaryotic stage in developing fluid bodies. Okay, so we think that the eukaryotic phase is spatially and temporally limited during the life cycle of the truffles. Then ascocarp will develop that the ascocarp is mainly made up of uniparental hyphae of uniparental origin. So, in summary, truffle life cycle is diploid. Tuber are generally heterolalic organisms. Mycorrhiza serves as a maternal partner in the cross. The soil mycelia are more dynamic than root resident strain. I don't have time to explain this. The distribution of sex is skewed in both natural and cultivated tuber melanosporum grounds. And I saw just some result of tuber magnatum. It's possible to type natural population tuber magnatum, tuber melanosporum according to molecular markers, according to the origin. So the main messages will be, we show that genetic competition occurs within tuber melanosporum strain for the persistence on the roots. The distribution of part of tuber melanosporum mycorrhiza is related, according to our opinion, to allelic configuration of the mating type of locus. So we can pursue now alternative strategy to set, at least as a pilot experiment, new truffle orchids. I leave this out, sorry. Just to, I would like to thank my collaborators, Andrea Rubini, who did most of the work, and for his brand wave, Claudia Riccioni and Beatrice Belfiore, she is our postdoc. And of course, I would like to thank Francis Martin and Claude Murat for sharing with us the results and approach and so on. Thanks for your attention.
Just a short question. Carlos.
Do you find that it's always the same maple type that is dominant? Like it's always MAC1 dominant, or it's always MAC2, or it changes with location?
No, I wouldn't say that. I mean, we don't have enough data to say that, but we think that, no. But we are testing this hypothesis. So we need, to increase the sample. Thanks.
I'm sorry, last question, Christine.
Looking at all these distributions and skewed distributions and alternatives for truffle culture, what about the role of animals in dispersing spores in, the way that it evolved?
So, yeah, you're right. I forgot to say that also animals, macrophages and animals, play a crucial role in the spreading.
So how would you include that factor in alternatives to truffle management?
I didn't think about it, honestly.
Thank you very much. I think we have no more time. So thank you, Francesco. The next speaker, bueno, tenemos un pequeñito cambio en el orden de presentación. Vamos a retomar la presentación que no se pudo hacer el día anterior sobre the role of irrigation in the root of truffles in Western Australia, que presenta Harry Slitch. Harry, it's time.
Thanks very much, and special thanks to the organisers for having me on not once but twice in the program, so I'm very grateful. Yeah, my name's Harry Eslick, and I work with the Wine and Truffle Company in Western Australia, and also I'm doing my PhD at Murdoch University. I'm going to talk about a small part of my research looking at the role of effective irrigation on the susceptibility of truffles to disease. So, just to repeat myself briefly, Wine and Truffle Company's plantation is down in Manjimup in southwest Western Australia, and this is Australia's most productive truffle producing region. So out of Australia's 4.5 tonnes which were produced last year, Manjimup produced 3.1 tonnes, and the Wine and Truffle Company accounted for 2 tonnes of that. But in addition to the 2 tonnes of truffle that were produced, there were another tonne of truffles that were produced but actually went rotten before harvest, so they weren't didn't make it to sale. But this figure, so that was 30% in 2012, but this figure's been up at over 50% in the last 4 years. And it was actually, since the beginning of production, it was increasing year on year until the start of my research. But this situation is not unique to the Wine and Truffle Company's property. In Australia, we know at least 3 of the top 5 truffle producers are having significant problems and losing a lot of truffle. So what exactly is going on? This is just some examples of truffles going rotten, and looking at it, there's a huge variety in the symptoms, so it's not really clear that there's one single biotic cause. There's kind of dark pigmentation in immature truffles, but then you get complete loss of melanisation in mature truffles, and you get different types of pigments occurring, pinks and yellows and things, and then you've got completely different dry type of rot in other truffles. That's just some more examples. So there's a huge variety of symptoms. But what is kind of common to all of them, or most of them, is that there's a significant relationship between the depth at which truffles are forming and the proportion of truffles that are going rotten. And a lot of our truffles are in this top 5 centimetres, most of them. And so what we think is happening is that truffles at the surface are becoming stressed by a whole variety of sources, such as desiccation, sunburn, insect damage, fungal pathogens, bacterial pathogens, and that eventually stresses them to the point in which they go rotten. So just briefly, the study site was established in 1998. It covers 20 hectares, and it's 90% Corylus avellana with mixed oak species making up the rest. It's in a relatively high rainfall zone, but it's very seasonal distribution, so most of the rain falls in the winter, and it's very dry summers, and deep, loamy, sandy loam soils. So since establishment, the plantation was irrigated on a weekly basis at a set rate of 20 millimetres per week. And looking back at the literature, this seems quite a lot more than what's usually applied in Europe. So, I mean, recommendations range from every 10 days to once a month in terms of irrigation. So we thought this would be interesting to look at. And this just gives you an example of the property and the extent of the problem. So one of the things we've been doing is covering the truffles that form too close to the surface with a bit of soil. So you can just see how many truffles are being produced and how many are really close to the surface there. So our hypothesis was that the high rate and frequency of irrigation decreases the depth at which truffles form, and this leads to truffle rot. So we designed an experiment. So this is a map of the property. Doesn't turn out too well, but each square here represents a tree, so there's 13,000 trees, and each dark tree is a tree that produced a truffle in 2011. And this is the experiment here. It was a split plot design. We tested 2 different rates of frequencies of irrigation, So either monthly irrigation or irrigation once a week, which is the standard, and then 4 different rates of irrigation. And the rate of irrigation was based on a proportion of estimated tree water use. So using reference crop evapotranspiration and a crop factor, which is similar to the method described on Tuesday. So the 4 rates were replacing 100% of the estimated water use, 50% of water use, 9%, or nothing. And the 9% treatment was supplied via drippers, so 2 drippers on either side of the tree. And the rest was a micro-sprinkler system, which you can sort of make out in the background there. But the experiment was repeated for 2 years. So in the second year, the 2 really low treatments were replaced by 37% and 50%. Okay, so to harvest the truffles, the property was searched on a weekly basis. And for each truffle, the harvesters had a little iPod with some remote databasing software. So for each truffle, they recorded the location of the truffle based on the nearest tree, the depth of the truffle, the size, and the condition, so whether it was rotten or not. So in 2011, there were 7,863 truffles produced, and all this data to analyse the effect of treatment on the truffle condition, we used log linear modelling. So in the model, we included irrigation interval, irrigation rate, truffle depth, and truffle condition for each truffle. And so the modelling gave us 4 significant interactions between this data. And I will explore that in a bit more detail in the graphs. So the significant interaction between irrigation interval, irrigation rate, and truffle condition showed that the two low irrigation treatments, 0% and 9%, gave a higher proportion of truffles going rotten only when applied on a weekly basis. And this is a little bit confusing because the weekly and monthly treatment when applied at 0 rate are essentially the same. So it's a bit difficult to interpret it, but it does show that perhaps there's more rot occurring in these low treatments, and this is the opposite to what we'd hypothesized. But it's potential that they're... Okay, so there's also this significant interaction between truffle depth and truffle condition, which we've seen earlier. And there's also, but this is what's really interesting, there was a slight increase in the proportion of truffles forming at the surface with the 100% rate treatment and the weekly irrigation treatment, which is what we'd hypothesized. So we did the experiment again on a second year, and we did the same modeling with the new data, 11,000 truffles. We got 3 significant interactions. And so similar to last year, there was a significant interaction between interval rate and depth. So the 100% rate treatment gave an increase in the proportion of truffles forming at the soil surface, but only when applied at weekly intervals. And also this 100% rate treatment showed an increase in the proportion of truffles going rotten. But what is quite interesting in this interaction between interval depth and truffle condition, when irrigation is applied monthly, you get this kind of standard interaction where truffles at the soil surface are much more likely to go rotten. But when irrigation is applied weekly, there's the same proportion of truffles going rotten at the soil surface as below the soil surface. So we think there's something else going on here. Potentially, soil hypoxia is a different cause of rot, causing the rot of truffles below the soil surface. So, just to conclude, the high rate and frequency of irrigation can decrease the depth of truffle formation, resulting in high incidence of disease, which confirms our initial hypothesis. But we don't fully understand the mechanism. So, is it that luxuriant supply of water stimulates truffles to form closer to the surface, or potentially is it hypoxia, which is preventing truffles from initiating lower down? And what's the role of root depth? So irrigation is stimulating the roots close to the surface, but we do have good mycorrhizal colonization down to at least 30 centimeters, but all our truffles are forming in the top 5 centimeters, so it's not purely an effect of root depth. And thanks very much.
Thank you, Harry. Volvemos al orden que correspondía al día de hoy, esta mañana, y la primera intervención será a cargo de Ismail Sbisi sobre desert truffles in Tunisia, diversity and cultivation attempts.
Thank you, Mr. Onrubia. So I have the pleasure to present you our work entitled Desert Truffle in Tunisia, Diversity and Cultivation Attempts. So the desert truffle are the fruit body of Sommiposium symbiotic fungi belonging to Ascomyza. They form in natural condition an ectomycorrhizal symbiosis, but this association can be influenced by external condition, especially the phosphor contents. So the genus Tirfesia, Tirmania, and Picoa are displayed in Tunisia. They are found also in North Africa, Middle East, Turkey, southern France, Italy, and Spain. Some species like Tirmania nivea have an antibacterial and antioxidant property. They are rich in protein, amino acids, fiber, minerals, and carbohydrate. And the ICOS extract of Tirfesia budiri are used in traditional medicine to treat a disease like blepharitis and conjunctivitis. So the desert truffle have or present a common ecological feature like a sandy soil with a pH close to neutrality, low mineral levels, and an alternance of seasons with evenly distributed rainfall, and the high density of host plant. Usually the desert truffle form an symbiosis with some species of the Cistaceae family, especially with the Cistus and Eleontimum, and occasionally with the Pinus, Quercus, and Cobrecia. So traditionally, the desert truffle of the genus Picoa, Tirfesia, and Tirmania have classified among the Tirfesaceae family, but recent molecular phylogenetic study have classified this truffle among the Pisizaceae. So in Tunisia, we have described the 6 species of truffle. Tirfesia budiri, Tirmania nivea, Picoa juniperi, Picoa lefevri in southern Tunisia, Tirfesia claveri in the center, and Tirfesia arenaria in the north. All these species form a symbiosis with Eleontimum sicilioflorum, except for Tirfesia claveri that form a symbiosis with Eleontimum almerans and Eleontimum caericum, and Tirfesia arenaria with Eleontimum guttatum. So we have described the specimens of unusual truffle collected from the center-east of Tunisia in mixed stand dominated by Acacia cyanophylla. So the ascocarps of the new species are sub-epogeous to epogeous, totally enclosed by the peridium and covered with the embedded particle of substrate. The ascocarps are solitary or in a small cluster with 5 to 9 even more individual, and they are 1 to 4 centimeter in size with irregular forms. The peridium is glabrous, white to cream at maturity. So the gleba is initially white to cream, then brown when mature, partially filled with the palisade iminium of Aci and paraphyes. So the Aci is cylindrical the Aci is cylindrical with tapering style, double-layered, hyaline, thin-walled, and contain mostly 5 ascospores. The ascospores are ornamented, pale brown, at maturity, 13 to 35 micrometer of broad. The paraphyes has the same length or longer than the Aci, 3.5 micrometer in diameter, cylindrical, abundant, and dense. So some morphological features of the new specimens are similar to Tiberaceae species of Dinglia, Labyrinthomices, and Rhinolomices genus, but is closely related to the southern species Rhinolomices parvulus porus, initially described by Bayton and Majazik. So the sequence analysis of the LSU and internal transcribed gene of the ribosomal RDNA that are really distinguished this organism from the other Rhinolomices species. So the new truffle was found around Acacia cyanophylla. This plant native to the southwestern Australia was introduced in Tunisia since 1904 for the fixing of coastal dunes of sublimate area, and its introduction has been generalized to the whole country. So the new species was likely brought from Australia together with their host plant, and they seem to have facilitated the establishment of acacia plantation. This phenomenon has been reported in the Iberian Peninsula with other Australian species like Laccaria and Rhinolomices. We have the same example with Rillandiella periblina, that is described as a new species in Italy, although this genus is endemic to Australia. So, on the other hand, we have in the northern Tunisia an humid climate stage with the forest dominated by Curcus and Pinus halepensis, that may develop the forest truffle of the genus TUBER or other species. In parallel, an experimental truffle field was established between 2006 and 2007 in Redland Institute of Medellin, with Elianthemum siciliflorum as a host plant. The seedlings of Elianthemum were inoculated by the spore of Tirfisa budiri, the most appreciated species by the local population in gnotoxenic condition. So the first cultivated desert truffle were obtained 2 years of the planting date. We have 4 fruit bodies, but the plantation have lost their productivity in the following year due to the rainfall limitation. For this, we decided to install an irrigation system and achieve the ideal level of precipitation. For economic reasons, we will try with irrigation by tap water and rainfall and rainwater. And thank you for your attention.
Muchas gracias, Simé, por completo de la presentación. Pasamos a la siguiente presentación, que viene de Polonia, la profesora Maria Labrinovic, que va a hablar sobre Poland as an environment for truffles.
Buenos dias, good morning. Truffles have been of increasing interest in Poland from mycological, ecological, and horological points of view. In last years, however, in changing of economical, especially agricultural situation, farmers are looking for new ways of profitable land using. Easy contacts between the people from south and north of Europe are favorable for widespreading interest in trafficulture. How does it look, Poland, as an environment for truffles? We have the Tatra Mountains in the south and the Baltic Sea in the north. Not yet. This means variety of sites. Truffles, exactly Tuber estivum, occurs in our neighbor countries. We all knew about successful trafficultures in Sweden. In Poland, great interest was in 19th century. After this period, nobody was looking for truffles. Tuber estivum has been red listed in the IUCN category as extinct. The old map data on the occurrence of truffles were set together by Lubelska from Krakow in 1953. She mentioned 8 species of truffles, among them Tuber estivum from 6 localities on the basis of the papers by Kaspary, Błoński, Aleksandrowicz from 19th century. Some of localities indicated at the map were looking doubtful on sandy alluvial soils. Yet, according to Gérard Chevalier and other authors, the conditio sine qua non for truffles is calcium in soil. In Poland, we have it in the whole territory, but on different depths. There are several methods to adapt soil in contents of calcium and pH suitable for the cultivation of truffles. On the map, there are indicated calcareous regions. On these areas, our attention is focused on the research concerned. A really limited factor seems to be climate. The average annual temperature was 8 Celsius degree, and the sum of rainfalls 600 millimeters in the area of truffles occurrence in the period of last 5 years. Distribution of the main host trees for black truffles of Quercus robur, Quercus petrea, Tilia cordata, Fagus sylvatica cover the area of Poland, similarly to Carpinus betulus and Corylus avellana. Besides, the fifth parallel of latitude traverses the south of Poland as well as the north of France. These are good prognosis for producing the Burgundy truffles in our country. How were the truffles in Poland discovered? At first, rediscovered. At first, the spores of Tuber aestivum were identified in stomach of contents of the mice caught by zoologists in 1980 of the last century in Ojców National Park near Krakow. These small creatures show us where we should look for truffles. As a matter of fact, after 100 years, Tuber aestivum was discovered on 13 localities and Tuber mesentericum on 5 localities in 3 regions indicated at the map. This map corresponds very well with a geological map presented by Chevalier at this conference. Example of localities of black truffles. The most famous concern is the Kraków-Częstochowa upland. There are 2 different localities of both Tuber aestivum and Tuber mesentericum monitored by us. The first natural oak hornbeam forest nature reserve, and the second an oak plantation at present 50 years old. The second region is Przedbórz upland, and the third one is Kielce-Sandomierz upland. Truffles produce fruit body only in some places called hypogeous oases or nests, where ecological conditions are suitable. Similarly, only in some years the climatic conditions are named formation of fruit bodies. It is remarkable that the oases of black truffles were found in the areas intensively used by the people, sometimes along roads, tourist trails, and places tramped every day. Observation of a natural site supply valuable information concerning requirements of truffles. This knowledge is helpful when starting to establish a trafficulture. The picture shows different phases of creation of the first in Poland truffier in the Kraków Częstochowa upland, this historical event. Krzysztof Belowski, the owner, consulted his work with French specialists in scientific and practical aspects and has shown us his results. First attempts towards cultivation of truffles on an experimental scale were already made several years ago in vicinity of Lublin by Professor Dorota Hilszańska and co-workers from the Forest Research Institute in Warsaw. At the end, Maison de Truffe. An idea to organize the Truffle Information Center was born on the basis of an enormous number of questions from local people and people of Polish origin living in so-called truffle countries, especially those having pieces of land in Poland. We start with permanent truffle carpophores exhibition open to public. Personal contacts with the people make us aware how important is education in the process of introducing truffle culture. Thank you.
Thank you very much, Maria. Next speaker is Simon Regli. He will speak about the Burgundy truffle in Switzerland, its phenology and ecological requirements.
Thank you. I want to speak about the Burgundy truffle in Switzerland, and we want to know more about the factors involved in the fruit body formation and the growth of this species in Switzerland. This project is a collaboration between our institute and colleagues from the University of Freiburg in Breisgau. And the reason why we do this research, one of the reasons is this publication by Uli Stobbe, who found out that the Burgundy truffle is not at all rare in southwestern Germany, even if the species is on the red list in Germany and not allowed to be harvested. So he found more than 100 sites in the region in the southwestern Germany of the species. Then a second reason is the increasing popularity of truffling in Switzerland. And one indicator is the number of dogs, of truffle dogs. So the lagottos, there are today about more than 1,000 lagottos registered in Switzerland. So it's very popular to buy such a dog and to go out hunting mushrooms. Then, last but not least, the detection of the discovery of a white truffle recently in western Switzerland also increased the popularity. It was the first Tuber magnatum found in Switzerland, north of the Alps. So, we started the project with the aim to get new insights in the ecology, phenology, and the growth dynamics of this species, and to look at the eventual impact of harvesting. We selected 8 sites, Burgundy truffle sites, and started a monitoring every 3 weeks on these sites, so all the truffles are harvested on this site, and every fruit body was weighted, the maturity was determined, and a sample from each fruit body for DNA analysis. And in addition, we measure tree growth on these monitoring sites. So we installed dendrometers to measure tree growth with a very fine resolution, so we can measure also daily increments or movements of the tree. And we measure soil moisture and soil temperature. Additionally, we do some archaeological excavation, so we want to know more about the distribution or the localization of the truffles in the soil profile. Here are the first results of 2 years. So we started in March 2001. Here what is March 2002, and so 2 years. And what we can see, we have a high variation between the sites in phenology and also in productivity. There are sites with a productivity nearly all over the year, and there are sites with very distinct season of occurrence. But we can see that there is no decrease in production. There are high fluctuations, but no tendency of a decrease. Here is the mean fresh weight. So we thought that the size of the fruit bodies will decrease as a result of the harvesting, of the regular harvesting. But no tendencies, no trend can be seen in the data. If we superimpose all the sites, we can see that we have production all over the year. So we have peaks in July, we have peaks in October here, and again the next year peak in June and in October. And most interestingly, we have a gap in spring. So in spring, the 8 sites didn't produce fruit bodies. And this may be, the reason may be the physiology of the tree, so the utilization of the carbohydrates in this season for growth. Then the data, all the soil temperature and soil moisture and tree growth data. Unfortunately, I can't show you results because we haven't yet done these analyses. But perhaps we will find answers on the phenology out of this data, I hope. And we will continue these inventories, these monitorings on these 8 sites, and we will also enlarge the number of sites. We will start with 8 new monitoring sites in Switzerland this year, and we will also start with 1 monitoring site in Hungary, in the plantation of colleagues Oscar and Istvan this year. And some words to the archaeological excavation. So we looked for sites with truffle docks, and then we took the truffle docks away and we began to dig in the soil. So very carefully down into the soil, through the soil layers, because we wanted to localize the fruit bodies in relation to soil depth, to ground vegetation, and to soil chemistry and topography and so on. And we started population genetic studies. Here the first results. This is such an excavation site, and we see here about 20 fruit bodies, the red dots, they belong to the same individuum. Then we have some smaller individuals and 2 individuals, 1 fruit body individuals. So we will continue this research also with mycorrhiza samples and soil samples. Here is a collection of such an excavation at one single date. So we found 10 fruit bodies on these 3 square meters. And we can see that we found very little immature fruit bodies, less than 1 gram, and we found big ones, 100 grams. We found all maturity classes, and we found also one already decomposed fruit body. So that means that we find various development stages at the same point, and we conclude out of that that Burgundy truffle is growing continuously all over the year, except in spring. Thank you very much.
Thank you, Simon. The next speaker, next presentation, is Matthew Smith. Will speak about the high diversity and widespread occurrence of mitotic spore mats in ectomycorrhizal pesticides coming from Florida.
Thank you. Thank you very much for having me. Today I'm going to talk about asexual spore mats in ectomycorrhizal Pisizales. I'm from the Department of Plant Pathology at the University of Florida in the United States, a very well-known truffle region I'm sure that you know. That was a joke. So I know I won't get to this whole story in 10 minutes, so I just wanted to point out that this whole story and the recent research that we've been doing on this is now available. It's now online at Molecular Ecology. I also wanted to thank the 2 main collaborators that I worked with on this project. First is Roseanne Healy, who's here. She's the first author of the work, and she's a PhD student at University of Minnesota. And then Greg Benito, who is a postdoc at Duke University. We worked on this closely together. I also wanted to thank Dr. Jim Trappy, who was another co-author on this paper. He's been very inspirational to all of us working on truffles. So thank you, Jim. He encouraged me to come. This is my first time in Spain, and it's been wonderful. Thank you. So we all sort of have a classic story in our heads about truffles, and sort of that's sort of shown here, that you have the plant, and then you have the mycelium exploring the soil, and at the ectomycorrhizae, you have this interaction between the plant and the fungus, and then through processes that are still somewhat mystical, the fruiting bodies are forming near the soil surface. And we sort of know, because we enjoy the flavors and the odors of truffles, that we think that animals are involved in the dispersal and are important for a lot of species of truffles. But as you've seen, this sort of classic story is changing pretty quickly as we learn more about some of the truffles. And so especially the genome has told us something about the diversity. It's told us something about the outcrossing, and as you saw, sex is pretty important. But we don't really understand exactly how this happens. And you've heard a lot of talks that are sort of asking important questions about this. I think it's important to consider tuber within the context of the other fungi in the group, the Pisizales, many of which are ectomycorrhizal. So I think it's important to consider the ecology of the entire group and also the evolution. And as well, I think it's important to incorporate natural history observations about these fungi. And so I'm not the first one to make observations about asexual states in ectomycorrhizal fungi in this group. There's some information in the literature already about that. Here are 3 examples. Rulandiella, which is a truffle-like genus that's from the southern hemisphere associated with eucalypts and nothofagus trees, is shown to make these asexual states back in 1989. And then in 2001 and 2004, there was good evidence that these ectomycorrhizal truffle genera from the northern hemisphere, Pachyphlois and Tuber, were known to make these asexual states. And I want to point out that, as far as I know anyways, there are no reports of asexual states from basidiomycetes, and I think that's part of why these have maybe been underestimated or overlooked in the past. So another thing to consider is that if you look across the group, the whole entire order Pisizales, there are actually a lot of fungi in this group that have asexual spore stages, and the function of these is not totally clear in a lot of these fungi as well. But I just wanted to point out that here's 3 examples. The morels are known to have these asexual spore stages. Some of the saprotrophic Pisizas that can be problematic in nursery kind of habitats. And as well, a fungus which we call Phymatotrichopsis omnivora, which is sometimes called Texas root rot, is a big problem in the southwestern United States. It's a generalist pathogen that causes really big crop losses, especially in cotton, but in some other crops. And it belongs to the Pisizales, and it makes these spore mats on the soil surface. So when I moved to the eastern United States from the western United States, I visited forests that were totally different from the ones that I was used to. And as I was walking along trails in these forests, I noticed a large number of these spore mats, and I couldn't tell from the morphology what groups they belonged to, so I started collecting them. I often found them at disturbed areas at the edges of trails or along the edges of the forest, and I noticed that there was a wide diversity of morphology from these. And so I had a hypothesis after I realized that some of them belonged to the Pisizales, that asexual states of these ectomycorrhizal Pisizales might be widespread and common, and also belong to different groups within the Pisizales. And you can kind of see that here from the morphology. So Roseanne and Greg and I began collecting these over time. We got up to about 300 different spore mats. We used the ITS and the large subunit to sort of place them phylogenetically and make species groups or operational taxonomic units. And we found about 48 different species that were spread across 6 different lineages of Pisizales. And those are shown here on this sort of basic strict consensus tree of Pisizales with just some representatives. But the main point is that they belong to these different groups that are all ectomycorrhizal that have appeared to have evolved the ability to form ectomycorrhizae separately, but all belong to the Pisizales. So these are Hydnotria, the Fischerla group, TUBER, which is important for us, and also the Tophasia group, which is also important for our discussion, and then Pachyphlois and Hydnobolides. And if you know anything about the Pisizales, you'll know that within all these groups there are truffle-like fungi, and I think that this may sort of help us or give us insight into some of the mystery about these truffle fungi in terms of how do they actually get around. It may not just be animals, that some of these other spore stages that are cryptic could be involved. So, as I said, asexual spore mats had already been documented by Alexander Urban in 2004, 2 different species in the Puberulium lineage, and they are these sort of verticillate-looking forms. So our work really was just building on that in terms of the tuber group. We found another species in the Puberulium lineage up here in several sites in the US, and then we actually found 2 species in the Maculatum lineage. And I want to point out that Roseanne did some measurements of these to try to come up with an idea of the number of propagules that you might find. And she found, on average, between 1,000 and 2,000 spores per millimeter of sort of soil coverage. So this is a large number of spores, and I think could be biologically very important for tuber and some other groups as well. So I'm not going to go through all the groups because I don't have time, but I want to point out a pattern that we found in most of the groups where we found these asexual stages. This is for the genus Hydnobolides, which forms these truffles. Sometimes they're called the gristle truffles in the United States. And what I've done is this is an ITS phylogeny where I've color-coded all of the terminals. The ones that are fruiting bodies are shown in pink. The ones that were spore mat collections are shown in yellow, and the ones that were ectomycorrhizae from GenBanks are shown in green. And this was a pattern that was common in a lot of these, where basically they were all intermixed. And what we found was that there was usually a lot more diversity based on the anamorphic spore stages and also the roots. And we also found, like for Hydnobolides, there's only 2 described species, but we have probably 15 or more species just based on ITS sequences. So there's a lot of diversity out there, and these anamorphic spore mats can sometimes tell us about the diversity in a way that we can't get from the fruiting bodies easily. In terms of when and where we found these, These were found all over the world. I was able to visit southern South America and China and find these in a couple different forest types. In those, wherever we found ectomycorrhizal fungi in the Pis Isales, we could find these spore mats. There were records from Europe. Collaborators sent us material from Mexico, and we visited 10 states within the US and found these. So it's a widespread phenomenon across ectomycorrhizal-dominated habitats. And then within sites in Durham, North Carolina, where I was living at the time, we found these basically whenever there was enough water throughout the year and it was above freezing, we could find these spore mats in forests. Whereas the truffles really were restricted to the end of our summer and the beginning of fall season. And so these occur over a long period of time and seem to respond quickly to weather conditions. Within given individual sites, there was high variation, and actually I'm not going to go too much into detail about this because it's in the paper, but just to say that pine forests did not have very much diversity and didn't have this as much, whereas oak woodlands tended to have high diversity and high coverage. The main thing that we're really missing with this information is the spore mat function, and it may be different for different groups, but I want to point out we did some minimal experiments. We didn't get any germination from media, and we were not able to get mycorrhiza formation. And so our working hypothesis at this point, while we're still looking for the proverbial smoking gun, is we think that these are spermatia that are for outcrossing. Another thing that we notice is for almost all the groups, they're small and they have thin spore walls. So we think they're for sex, for outcrossing. So just to conclude, these appear to be common across the Pis Isales, and they include edible truffles, at least for some, such as Tuber borkii. The function is unknown, but we think that this is likely to be of the function of spermatia for outcrossing. And the last point I want to make is that we found that these really obvious forms were very common in the forest, but we think that these asexual spore mats may be a really cryptic hidden stage. And I want to point out that the ones we were finding were really obvious white mats in many cases that are pretty easy to see in a dark forest. But some of these other ones may form smaller ones that are ephemeral, and they'd be more challenging to find. So I had many collaborators who helped with this work, and I want to thank them, and especially the University of Florida for supporting my research. And lastly, I want to point out that I have two posters out there. One is Greg Benito is the first author on the biogeography and evolution of Tuberaceae, and another is on a truffle that actually does occur in Florida, the pecan truffle. So if anybody's interested, I'm glad to talk about those later. Thank you.
Gracias, Matthew. Ahora tenemos dos presentaciones seguidas que nos hará Alexander Urban, de la Universidad de Viena. Okay. La primera de ellas, inicialmente la iba a presentar Marianne French, y versa sobre Tuber mesentericum ascocarp development.
The presentation is fully. Okay. Perfect. Okay. Thank you. So for this presentation, I have no credits. It's all the work by Marianne French, who cannot be here, unfortunately. It's about ascocarp development and root dependencies in Tuber mesentericum. So I would like to first describe the methodology she used. Well, she just tried to make descriptions based on published criteria of ascocarp morphology and ectomycorrhizal morphology, and in a second stage, develop her own descriptive system to grasp some new features or newly observed features occurring during development. Okay. So the classic methods are in the literature, it's well known, and among the
what is an important observation, that there is morphological variation in Tuber mesentericum. So all of these observations of dependencies between morphology and weather conditions and developmental conditions have to be relativized, because there is a background of variation in the species between different origins, different specimens, and so it's sometimes hard to discern the variation resulting from genetic variation and resulting from environmental factors. Nevertheless, she characterized some patterns, like very small truffles without a cavity. Tuber mesentericum, you probably know, typically has this central cavity, very flat ascocarps, some variations in the shape of the cavity, a transversal cavity, and she regrouped these observations according to sites and according to weather patterns. And also she made observations on the attachments of the roots to the fruiting bodies. Probably you know that in Tuber melanosporum, the root is usually not attached to the fruiting body. So the connection is made by the mycelium only. And according to her observations, this is not the case in Tuber mesentericum, and I think it's an important information, because it's easy to reproduce these observations and maybe to know more about some ecological differences. It seems that the Tuber mesentericum is really very much attached to at least one root, which she calls the birth root. And maybe you know that in other hypogeous fungi, for example, in Elaphomyces, you can observe a very narrow attachment of roots to the ascocarp. So, for example, in some Elaphomyces species, the ascocarps is really wrapped in the roots. It's within a nest of roots. And so there seems to be some intermediate situation in Tuber mesentericum. And, well, I'll explain it further. So these are more or less typical situations in Tuber mesentericum. And the cavity, sometimes it can be less well developed, just a small depression. And what happens after dry spells is that you can find a lot of such, well, not decaying, but kind of obliterated fruiting bodies, and which are completely shrinked. And maybe this is part of the process described by Giovanni Pacioni with the soil radar, the occurrence of such obliteration. But interestingly, when rain comes back, you find also some special morphologies among those which survive. And for example, you can have this form, which you observed only after longer periods of drought, and also some more transversal cavities. Another observation is that sometimes small fruiting bodies are attached to a larger one. My personal opinion is that it's just a multiple birth of fruiting bodies from several individual primordia. But she thinks that it's kind of sub-cloning of this spore cup. Okay, then her observations on the roots. So there is one root which is quite tightly attached to the fruiting body, and you can even see some imprint of the peridium of the fruiting body on the root. And of course you see connections by mycelia. And interestingly, there grow roots into the cavity, but these roots, they are never closely attached to the fruiting body. So this is just, they just grow into the cavity incidentally. And the roots which are attached to the fruiting body is always occurring on the outer side, on the opposite side to the cavity. And this is maybe a bit counterintuitive, but it fits very well to our idea of the developmental process in tuber mesentericum. Okay, and so that's why she calls the roots which are growing into the cavity secondary roots. So it has nothing to do with primary and secondary roots, as usually said. It's only the birth root is the primary root, which is attached to the fruiting bodies, and the others, they are not as tightly attached to the fruiting body. It's more incidental. And yes, so these were the main observations by Marianne French on the development of tuber mesentericum. I'm not sure if I can really respond to questions because it was really her own observations. Okay. I hope I did it in her sense. Thank you. So what I want to present now, it's a work of many years together with many colleagues. Since actually several of us started to explore the diversity of small truffles independently, and so we more and more joined our data together and specializing on different groups of these small truffles. Tuber excavatum is one of the non-commercialized truffle species, and so why should we deal with it at all? Because it's there. It may have some features, some ecological and biological features which are different from other truffle species. So to my observation, it seems to be less frequent in typical pioneer situations and more in the more mature forest or bush ecosystems. And it may accompany or possibly replace other truffle species during later successional states, and especially when the environment gets more closed. This is our observation. Sometimes we find it at the margins of truffle beds where the environment is just more closed. And according to my observations, it's also fairly drought resistant, so you can find it in seasons when you will find little other species because it was too dry. And it's a very common collateral find when searching for gourmet truffle species. So there must be lots of material already been seen by many of the truffle researchers, or also the people who search for truffles. And commonly in the literature, and also in the recent taxonomic revision by Cerruti, there are two species recognized as species. And the one is Tuber excavatum and the other one is Tuber fulgens. So this is a more or less typical specimens of Tuber excavatum. And on the right, I show you two different collections of Tuber fulgens. The upper one is from around Vienna, and the lower one is from the Pyrenees, in the Pyrénées-Orientales in France. And the distinction is based on the one hand, based on color, and on the second hand, it's based on microscopy. So you have on the left side typical Tuber excavatum spores, which are more elongated, and on the right side typical Tuber fulgens spores, which are more globose. And also the volume is a bit different. Spore volumes are different, and also the distribution of number of spores per ascus are different. And well, we sampled as many specimens we could from as many countries where we had specimens available, and did some phylogenetic analysis. And well, the tree which you see here is really, I saw it only this morning, that it's not readable at all with this presentation, so I will open the file, the original file, and scroll through it a bit. So I hope you can see it better now. So what we see is that we have one grade which includes most of the tuber fulgens specimens, which however are distributed across numerous subgroups. And then there is one clade which includes most of the excavatum specimens originally identified as tuber excavatum. So it seems that this basic distinction between tuber fulgens and tuber excavatum is more or less valid. There had been some, so this distinction had been challenged already at all. And however, there is enormous phylogenetic structure within the group, and also the genetic distances are far beyond what is common in ITS sequences of ectomycorrhizal fungi. So the genetic distances are reach almost 20% between some specimens. So it's really a huge diversity. Unfortunately, so far we have data from one locus only for a larger sampling. So let's come back to this this presentation. Okay, so we did the barcode analysis to explain the barcoding gap shortly. It means that the interspecific variation is hypothesized to be much inferior compared to interspecific speciation. And in many cases, however, we would see an overlap of both. And this was the result of an analysis done by a program developed by Pyandra et al. It was up there in the methods in 2012. It's called automatic barcode gap analysis. And it shows that there seems to be a gap somewhere in this region. So there is important distance between the less related species and less difference among the more closely related specimens. And this analysis also allows to make an hypothesis how many species are included in this sampling. And the default conditions resulted in 9 species. And then there are some recursive iterations of the method. And according to the prior, depending on the prior of interspecific variety, you would get a result between 8 and 36 species in the sampling. Of course, this upper value is very unrealistic, but I think everything between 8 and 16 species in this middle range of the prior is realistic. So we have to explore this much further to see how many species there really are in this group. And we also, these are some preliminary results on morphological analysis. We found that there is morphological variation in the spore sizes and mesh sizes and so on, and that some of the groups come out quite well, so they occupy a certain place within the parameter space. But unfortunately, the quantitative traits are overlapping to a very high degree, so it's very difficult to define species based on morphology. Even though the groups come out more or less separately in the character space. So the question is, are these good species? How many are there? And if there are so many species, what was the driver of speciation? And we will explore this further by including more data on biogeography, host specificity, more metadata. And finally, we have to make some taxonomic conclusion, but we are not yet there. There is a lot of varieties and also species names available in the older literature. And, well, the difficult task will be to assign the genetic diversity to the historic taxonomy, to make the link between both of them. And, well, I just have a request to all of you who are out there in the field. Please do make collections. Such a work is really, it needs a lot of collections covering a huge area, and especially in truffles, it's not easy to collect on your own. So we need collections. It's easy, and it's you find TUBER excavatum group very typically when you search for other truffles, and it's really valuable if you make such collections. Okay, thank you very much.
Well, the last presentation is Merenil from Hungary will speak about the phylogenetic analysis of TUBER brumale group.
Good morning. In my presentation, I would like to talk about our study of TUBER brumale, and our aims were to investigate the phylogeny, phylogeography, and the distribution of this species, or maybe species complex. But why have we chosen TUBER brumale? Because it is the only species from Melanosporum lineage which can be found in most of the European countries, and owing to its organoleptic properties, this truffle has by far one of the best economic values. On the other hand, TUBER brumale is one of the most frequent contaminating species in TUBER melanosporum and TUBER aestium orchard. And TUBER brumale is also non-native, for example, in New Zealand. Nevertheless, more and more is known about the biogeography of the genus TUBER. Only a few articles deal with phylogeography, distribution, and genetic variability of a single species. So this is why we started investigating with TUBER brumale. To cover the area of this species as fully as possible, we analyzed sequences from GenBank and more than 90 own sequences originating from 18 countries, mainly from Europe. And first we examined the phylogeny and the haplotype network based on ITS. After that, two other more conserved regions, the large subunit of ribosome and the protein kinase C, have also been investigated in a smaller subgroup. And the micromorphological measurements were performed on 52 specimens. And the results, this is a phylotree based on ITS, which contains the main lineages of the genus TUBER. And it seems that TUBER brumale is included Melanosporum lineages, and its motifs form two clade A and B. And the differences between the two clade, according to the nucleotide divergence of ITS, is about 10%. this is far over 3%, which is generally accepted level of interest-specific variability. And this is a phylotree, ITS phylotree also, which contains all of examined samples. And it showed that in the clade A, a new group is separated. So in the end, TUBER brumale formed 3 groups, which are symbolized by colors in order to facilitate understanding. Clade B is signed green, and the 2 groups from clade A are colored blue and red. The relationship of red and blue groups are not monophyletic, so this only represents 2 haplogroups. The large subunit of ribosome is much more conserved region, like the ITS. However, the separation of the 3 group is also shown in this tree clearly. Despite the conserved characteristics of LSU, it's linked with ITS. So to examine an unlinked region, like the protein kinase C, is necessary. In this tree, based on PKC, the red and blue samples are mixed, suggesting that the red and blue samples belong to one species, marked E, and the samples green belongs to another species, marked B. If you observe the occurrence, the 2 species, it can be concluded that there are 2 species with sympatric distribution. Furthermore, species B occurs only in the Carpathian Basin. Unfortunately, we didn't find any differences in the micro or macromorphological characters between the 2 species. However, we examined a lot of characters. This is the list. Currently, we are examining the structure of the peridia. In order to separate the 3 groups rapidly, we worked out an ITS-RFLP method, where the ITS region were cleaved by MBO1 restriction and the nuclease. And these are the patterns we get. In the rest of my time, I would like to present the phylogeography of the species A. This is a haplotype network where each shape represents a haplotype, and the lines between the shapes represent the mutations. The red and blue part of the network is equivalent to the red and blue groups in the ITS phylotree. So the red samples can be found almost everywhere in Europe and in New Zealand. And this part of the network indicates strong bottleneck effect and recent expansion. In spite of a smaller sample size, the blue samples indicate higher variability, and the border of their distribution is the Alps. This is more manifest if both are visualized in this map. We can see that the 2 haplogroups co-occur in the Carpathian Basin, and while only one of them is present in Western Europe and in the Balkan Peninsula. Therefore, we suppose that the 2 haplogroups survived on or found shelter in 2 different refuges during the last glacial. In summary, in contrast to previous publications working with small sample size, the ITS variability of TUBER brumale is much higher than was thought before, and it seems that TUBER brumale can be separated to species which have not been supported by morphological properties. Thank you for your attention.
Thank you very much, all the speakers. Now we have only time for 2 questions. Yeah.
A ver, me llamo Víctor Pérez y la pregunta es para Francisco, el doctor Francisco. Aquí hay muchas personas que no tenemos el nivel científico como ustedes y según su teoría, si no lo he entendido mal, seríamos capaces de producir trufas sin en árbol, ¿no? Sexualmente.
I don't think so. Not yet, at least. Yeah, that's right.
Just one idea. In the last presentation, it was very interesting. But I'd like to suggest that probably the differentiation didn't take place in the last glaciation, but perhaps in the Pliocene era, because there is a strong differentiation between the Pannonian Basin, so from the Miocene, okay, and the Western Europe. So maybe this differentiation can be much ancient than you think.
Thank you.
I think this would also fit with the relatively high differentiation of the high distance in the ITS region.
Last question.
Hola, buenos días. Soy Joaquín Doñate. Quería preguntar sobre la última ponencia. Si han podido averiguar cómo llegó la espora de TUBER brumale a Nueva Zelanda, si llegó en barco o fue andando o estaba ya allí. Gracias.
Unfortunately, we didn't examine the samples of New Zealand.
Ian Hall, New Zealand. It seems highly likely that the early introduction of brumale into New Zealand was as a contaminant in small cracks in the surface of melanosporum. We did our best to sanitize the truffles, but in those days we had no DNA techniques, so it was a mistake which was inevitable sooner or later.
Bueno, pues terminamos así, de esta manera.